Hey guys, Mr. Hyatt here. This is the end of the Chapter 16 lecture. We're talking about DNA errors. We're talking about mutations, things along those lines. So you can see on the screen that our mutation rate is about 1 in a billion base pairs, which means every time our DNA is replicated, we make about 3 mistakes. So that's pretty accurate. Why is that accurate? Well, our DNA polymerase 3 is our spell check. It's going to go back through and it's going to identify mismatches. Then there's a kind of a whole family of enzymes that are going to come in and kind of help with that spell checking process. Notice here we've got over 130 different DNA repair enzymes known. So it's something that is important for us to control. So we spend a lot of energy, a lot of resources, and a lot of time making these specific enzymes. Obviously, if you can't repair mutations, you're going to lead to things like cancer and other health issues. An example is this Xeroderma Pigmentosum, which is essentially where a DNA repair enzyme doesn't work, which leads to a lot of skin cancer. So you'll see that in the next slide. So there's an example of someone who has the cancerous cell and someone who does not, someone who's normal or has the wild type phenotype. We see these things called thymine dimers. That's going to be where we get thymine. So there's a bubble. We've got too many rings in our strand of DNA. So that's an issue. These are often caused by UV radiation. Those are often the mutations that you get. Maybe you get a really bad sunburn. You don't get cancer. You get a thymine dimer. Our body can excise that, can take it out, and repair it. That's sort of where we're headed with this slide. We've got this thing called DNA polymerase. That should sound familiar. Polymerase is going to build the new bases. It's going to fill in the new bases. We're going to have an enzyme that's going to cut the damaged DNA out, and then ligase is going to connect it. So here's a picture. We come through. Nuclease is going to remove. Then we're going to bring DNA polymerase through. It's going to add the correct bases back in. And then ligase glues them all together, kind of connects all the bonds. But we've got some issues with our DNA replication. We can only go 5' to 3', so that means the very end we can't do anything with. The very end of the strand we can't duplicate. So that means every round of replication our DNA is going to get a little bit shorter. So here's kind of what we're talking about. We've got this RNA primer here. Since we can only go 5' to 3', we're a little bit shorter. The next round, we're going to be a little bit shorter even still. And so each round, we get a little bit we get a little bit shorter, a little bit shorter. We can use these as kind of an internal clock. We have these things called telomeres at the end of our chromosomes. You can see on the screen, they're between 100 and 1,000 repeats of TTAGGG. Kind of filler DNA. We're going to talk about what it appears they might do here in a few slides. But basically it's cushion. The telomeres shorten with each DNA replication. That doesn't severely impact our systems, so that sort of allows us to not lose important DNA. When we die from old age, if it's truly from old age and it's not just because your immune system is weak and you die from pneumonia or something like that, when we die from old age, it's because our telomeres have gotten too short. So again, we can use it as sort of an internal clock. Here you're seeing the telomeres lit up on the screen. So you can see at the ends of the chromosomes, we've got telomeres. At both ends of the chromosomes, we've got telomeres. Also notice how few of these look like V's butt to butt. Maybe that one. But a lot of these are chromatids. They're telocentric, so they're connected at the end. We don't see a lot of those, so I wanted to point that out. Kind of have already talked about this. When the telomeres are too short, the cell dies by apoptosis. You should recognize that term at this point. If you don't, look it up. Obviously, if they're getting shorter, that's going to limit how many times the cell can divide. That we're essentially establishing a lifespan, a life limit on cells, and you can extrapolate that up to organisms. There is an enzyme called Telomerase that's really active in cancer cells that rebuilds those telomeres. You may have heard of HeLa cells, Henrietta Lacks cells, that they're treated with a lot of telomerase, and essentially it makes the cell immortal. Now, it doesn't make it immune to damage. You could still dump hydrochloric acid in a thing of HeLa cells and kill them, but they're not going to die from natural causes. They're not going to just run out of replications. Limited activity in cells like skin cells. We find them in germ cells, so as babies are developing and things like that. But could it extend the human life? Absolutely. Telomerase could come in and rebuild these telomeres and could extend our natural life. That doesn't help us if we're hit by a bus or something like that. But it could extend our natural lifespan. Could stop cancer if we could turn if we could turn off telomerase in cancerous cells. Maybe we could just turn off mitosis in those cells, and then cancer is no longer a problem. Or we don't have to worry about turning off mitosis because the telomeres just get so short that the cell dies from apoptosis. So, check this out. The DNA of telomeres, sorry about the typo there, is actually used to build proteins. These proteins seem to impede telomerase, so that should sound like a feedback loop at this point. So when we lose the DNA, we stop being able to impede telomerase. Now, we don't express telomerase very often, so in most conditions, that's not, or in most conditions, in most of the time, that's not an issue. Last thing in this section is how we wrap our chromosomes. We need to focus, like it says on the slide, on nucleosomes and chromosomes. Here's a picture. Okay, so we've got our DNA down here. Think of the demonstration I did with my yellow rope where I wrapped it around my leg. So we've got our chromatin. It's a double helix. It's soupy. Looks like spaghetti. Then histones come in, and that's my arm. The histones are going to spin and wrap the rope up and organize it. Then our histones are going to come together and form the nucleosome. I've got a little bit more after that. Nucleosomes are sometimes called beads on a string because that's what they look like. It is DNA wound on a protein core, so it's that protein histone that wraps the DNA around. It does control gene reading because it basically controls where the DNA resides when it's packed in a chromosome. A couple things here I'm going through quickly because we don't really need to concern ourselves with them. But those chromatin fibers are going to be, we could connect the last picture and this picture. So we could go from here and connect this part with this part. That's kind of what the idea here is. So the 30 nanometer fiber is thick, and then we loop the 30 nanometer fiber together, and that forms our chromosome. So that's sort of where I put my leg through the rope. I could have looped it around my leg to make it a little bit more accurate, but sorry. We know about chromosomes, so that shouldn't be anything new. So yeah, that is the end of the Chapter 16 lecture. Make sure you know the scientists and roughly what they did and what they learned. Make sure you can explain why DNA is an excellent genetic material. Make sure you can explain why DNA is a better match for our experimental results than is protein or RNA. You need to know how DNA replicates. You need to be able to identify problems in replication, so mutations. And then you need to explain the last few slides on chromatin packing.